drug is related with the biological activity. That means you want 100% biological activity means which are the groups has to be substituted. The groups which has to be substituted can be followed by a graphical or mathematical relationship. So you will be having again number of equations based on the fragment or the lead you are taking. If you are taking the lead like benzoic acid, it's a small moiety which acts as an anti-infective agent. If you substitute a halogen group, that is electron withdrawing group, the benzoic acid activity, monochlorobenzoic acid activity will be compared with the unsubstituted benzoic acid. So you will be calculating the biological activity in terms of substitution by the help of a mathematical expression that is called as quantitative structure activity relationship. I will define once again. The quantitative structure activity relationship is how much amount of quantity, that is biological activity, each group is going to give to the compound. That means you are having unsubstituted benzoic acid, you are having a substituted benzoic acid. The contribution of the X by various substituents is done in a computer in a two-dimensional way. So you will get the biological activity. If the biological activity is higher in case of one substituent, that particular component will be synthesized in the laboratory. That means your earlier days in the conventional method of synthesis, you will be preparing a salicylic acid and you will be testing in the animals or you will be testing in the cell lines. Nowadays, we are, due to the animal experiment like restriction, IAEC, we are going for hyphenated techniques. One among this is quantitative structure activity relationship, where the receptor, how it binds with the drug, is calculated mathematically. So what is quantitative structure activity relationship? It is a mathematical expression which how many groups are there, that groups, how much it is going to contribute to the biological activity. So it is an assumption. So it is not a practical. You are going to find out n number of compounds and in this n, which is the best compound. You are going to devise an equation. So finding out the best fit equation is the challenge. So the challenge in the quantitative structure activity relationship is finding out the best fit equation. So once the best fit equation you are finding out, you can go to the laboratory. You can see the stereochemical interpretation. You can go to the synthetic moiety and you can refer the synthetic schemes and you can develop. So this helps the scientist to come out with a lead by avoiding the practical experiments. That means, what are the advantages of quantitative structure activity relationship? Means time is saved, the chemicals are saved. After finding out some n number of experiments, you are telling that the biological compound is ineffective. You are wasting that much of chemicals. Environment friendly because you are not throwing any toxic chemicals. So this particular drug design techniques are also called as green chemistry. It comes under the topic called as green chemistry, where the toxicants or the chemicals are reduced or minimized because you are going to synthesize only the needed or already calculated compound. So this type of quantitative structure activity relationship is the role, the scientist role or the mathematician role is to understand the biology. So the pharmacists are signing in QSAR due to the two factors. One is we are learning the body structure, functions, everything in detail in human anatomy and physiology. So the biologist is well known for the pharmacist. And the next one is we are able to interpret with the pharmacological activity. So this successful interpretation of a pharmacist earns lakhs and lakhs. So the advantage of the drug design is once you are interested in computational techniques, tools, you may also come out as a computational chemist. That is the aim of QSAR. That is the basic topic in computer aided or drug discovery techniques. various approach la ஒரு approach வந்து QSAR. சரியா? This is also like in 1990s, late 1990s it was introduced and many scientists have saved the practical cost of the experiments, thereby the moieties which are synthesized in the laboratory are proved biologically active. So the advantage is number one is it reduces the experimental cost. Disadvantages, you cannot imagine the three-dimensional structure of the receptor through the equation. That is the disadvantage. Some of the equations are failed because inside the body the three-dimensional structure are sometimes cannot be predicted exactly. That's why we are having some pitfalls in QSAR. Advantages, green chemistry. Disadvantages, you cannot predict the receptor site exactly. That is the